Thank you for viewing our poster. I'm John Lambert and I'm the CEO of Amplia Therapeutics. Amplia is a publicly listed company based in Melbourne, Australia, and we're developing small molecule inhibitors of focal adhesion kinase, or FAK. If you have any questions about our presentation, please email them to me using the address john@ampliatx.com. FAK is a non-receptor tyrosine kinase with key roles in a variety of cellular processes, in particular those related to the adhesion and migration of numerous cell types. At Amplia, we believe that a FAK inhibitor with a suitable safety profile ought to be developable in both oncology and fibrosis indications. And so that we could address both these opportunities concurrently, we decided to initiate clinical development of our FAK inhibitors with a phase 1 trial in healthy volunteers. Amplia's lead candidate is called AMP945. AMP945 is an orally bioavailable small molecule inhibitor of FAK, and in vitro it potently inhibits FAK with an IC50 of about 2 nanomolar. And in a panel of 468 human kinases, AMP945 was shown to be highly selective for FAK. Our preclinical studies also tell us that AMP945 does not inhibit or induce cytochrome P450 enzymes, meaning that it has a relatively low risk of interaction with other medicines. Working with our collaborators, we have shown that AMP945, when given as pulsed doses and when added to nab-paclitaxel and gemcitabine, extends survival in an orthotopic mouse model of pancreatic cancer by around about 33%. Based on the preclinical data package and completion of a suite of safety studies compliant with the requirements of ICHM3, we initiated a phase 1 trial of AMP945 in healthy volunteers. The trial was a double-blind, placebo-controlled study assessing single and multiple ascending doses of AMP945. Cohorts of 8 volunteers were randomized in a 3:1 ratio of AMP945 to placebo. The effect of food on absorption of AMP945 was tested in the 30 milligram single-dose cohort. Plasma and urine pharmacokinetics was assessed at serial time points, and paired skin punch biopsies were collected before and after dosing in selected cohorts. Further information about the trial is available on the Australian New Zealand Clinical Trial Registry, or ANZCTR, with the registry number shown at the bottom of this slide. Orally administered AMP945 provided excellent plasma exposures of AMP945, with exposures after single and multiple doses being generally proportional to the dose given. Half-lives of AMP945 ranged from 15 to 23 hours, indicating that once daily dosing is feasible. And volumes of distribution also suggested that AMP945 is widely distributed in tissues. During the trial, we developed and validated a sensitive assay to measure phospho-FAK, the active form of FAK, in skin punch biopsies. Paired skin punches were collected from volunteers both before and after oral dosing with AMP945 in the 125 milligram single dose cohort and in each of the 3 multiple ascending dose cohorts. In the multiple ascending dose cohorts, skin punches were collected before dosing was initiated and after dosing was complete on day 7. We analysed the data by looking at it in 2 ways, both looking at the percent change in phospho-FAK from baseline, either by dose or by plasma area under the curve achieved for individual volunteers. In both analyses, we saw a clear trend in decline of phospho-FAK related to dose or exposure of AMP945. In the trial, AMP945 also appeared to be safe and well tolerated at all the doses we tested, There were no serious or severe adverse events or study withdrawals. There were no dose-related trends in clinical signs or safety labs. An AMP945 safety profile appeared indistinguishable from placebo. Overall, we found AMP945 to be a highly potent and selective inhibitor of focal adhesion kinase. And in the trial we're reporting today, it was safe and well tolerated across single and multiple ascending dose cohorts of healthy volunteers. The pharmacokinetics and pharmacodynamics of AMP945 demonstrate that it has wide tissue distribution, a half-life that supports daily oral dosing, and engages with its intended target. We saw a predictable dose-exposure relationship, as well as evidence of a pharmacodynamic effect in skin punch biopsies. This data supports continued development of AMP945 in patients with solid tumours and fibrotic diseases. And in fact, given the excellent pharmaceutic, safety and preclinical efficacy data we have in hand now for AMP945, Amplia plans to initiate a phase 2 trial of AMP945 in combination with nab-paclitaxel and gemcitabine in first-line patients with advanced pancreatic cancer in the very near future. We will report our clinical efficacy data as soon as available. Thank you for your time and attention today.